Hello, chemists and biochemists. I hope you're having a good day. What I'm going to talk to you all about today is enzymes. A little bit more about enzymes. What I want to get into today is the classification of enzymes and the nomenclature and the kind of identifiers that are associated with different enzymes. Now, historically, there have been a total of 6 different groups that every single enzyme that is known to man is going to fall into. Now, as of 2018, there's a total of 7 different groups. This doesn't radically change the first groups, but it helps classify some enzymes a little bit more. Now, for every single enzyme out there, there's going to be a numbering scheme that's going to look like this. X period, X period, X period, X. Okay. Now, each of these X's corresponds to a digit. Now, for the first one, it's going to range from 1 to 6. And this is going to be your enzyme's class. The second digit here, that can range, and it's variable based on what 1 through 6 is, because there might be more what are known as subclasses for different enzymes. So the second digit is your subclass. The third digit is going to be your subclass. And then finally, the last number here is going to be your specific enzyme. So it's pretty common to see something, for example, like 1.1. Let's go with 7.295. Okay. So all of those digits are going to give you some information about what that enzyme does until ultimately this last digit, this 295, as an example, would be the specific enzyme. Now, this 1 through 6 are class. 1 through, let me change that actually to 1 through 7. Those are going to be the major groups and classes of enzymes. So your first group are your oxidoreductases. Now, these are enzymes that are going to be involved in oxidation and reduction reactions. So one species is going to be oxidized and another species is going to be reduced. Your next group, or your next class, is going to be the transferaces. When a group is transferred from one species, from one molecule onto another molecule, so a functional group, like the carboxylic acid is transferred from one molecule to another molecule. Number 3, those are going to be your hydrolases, where we are going to use water to basically break a bond and remove a functional group or remove a molecule or a portion of a molecule. A lyase, lyases are group 4, where these are going to add or remove groups to form double bonds. Your 5th are your isomerases, converting from one isomer of a molecule to another isomer molecule. 6 are your ligases. These are going to be molecules where you're harnessing ATP's energy to basically combine 2 molecules together. This is an enzyme that's going to take 2 molecules, ATP, and bring those 2 molecules into one and break ATP to drive that. Your last group are known as your translocases. So this is the class of enzymes that is involved in movement of one molecule, perhaps an ion or something like that, from one side of a membrane to another side of a membrane. So those are your 7 different classes. Now, every single enzyme that is known is going to fall into one of those 7 categories. So to get into each one of these individual classes a little bit more, it's going to take a little bit of time. But the first one that I'd like to get into are your oxidoreductases. Here you can see an example of an oxidoreductase. This enzyme is known as lactate dehydrogenase. And what lactate dehydrogenase does is it takes this molecule right here, lactate, as well as nicotinamide adenine dinucleotide, and produces pyruvate and NADH. Okay. Now, one good, I guess, sort of tell or indicator that you're dealing with an oxidoreductase is that you have something like NAD plus and NADH. Those are the oxidized and reduced forms of nicotinamide adenine dinucleotide. So all that we're doing is we're taking this hydroxide group and this hydrogen, and we're producing a double bonded oxygen on that carbon right here. So this is a good example of a oxidoreductase. Next up, we've our transferaces. So these transferaces, remember what we're doing is we're transferring a portion of a molecule onto another molecule. So transferaces are the types of molecules that you're maybe a little bit more familiar with as soon as you start talking about a pathway like glycolysis. Now, what I have here on the left-hand side is I've got glucose, the cyclic form of glucose, and I've got ATP or adenosine triphosphate. Now my product is glucose 6-phosphate, or which I'm going to label as G6P. My other product is adenosine diphosphate. So we're taking one of these 3 phosphate molecules from adenosine triphosphate, and we're taking this OH group on glucose and replacing it with OPO3. So the phosphate from ATP is moved to glucose, specifically onto the 6th carbon of glucose, leaving behind ADP. So the key there is the molecule ATP donates a phosphate group to glucose. Glucose accepts it, making glucose 6-phosphate and leaving behind adenosine diphosphate. A large number of enzymes fall into this category where they are represented, or they're in the class of enzymes known as hydrolases. But one that might be relevant to anyone who's involved in beer brewing is an enzyme known as maltase. It takes this disaccharide known as maltose and uses water to break it into 2 separate glucose molecules. Now, another enzyme that you may or may not have heard of that's also classified as a hydrolase is RNase, RNase A, the molecule that takes a single-stranded RNA molecule and breaks it up into individual pieces. The key is that a hydrolase is going to use water in order to generate 2 products. Our next class of enzymes is our lyases. And what lyases are going to do is they're going to generally break a carbon-carbon bond, a carbon-nitrogen bond, carbon-oxygen bond. There's tons of different subclasses of lyases. One specific enzyme is an enzyme aldolase, which is commonly found in glycolysis. And what this enzyme does is it takes this 6-carbon molecule, this fructose 1,6-bisphosphate, and it's going to split it by breaking this bond right here and this bond right here. And it's going to make dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. So taking one 6-carbon molecule, a 5-membered ring, and splitting it into 2 equal 3-carbon molecules. So each one of these, just for perspective, each one of these 3-carbon molecules ends up with a phosphate. But the key here is that lyase is doing this, and it's doing so without any sort of additional water. It's breaking these carbon-carbon bonds, this carbon-oxygen bond, without any sort of water to drive the reaction. The next class of enzymes, the isomerases, actually kind of in the context of glycolysis, one example of an isomerase is you see the 2 molecules that are the result of the enzyme aldolase, where dihydroxyacetone phosphate and glyceraldehyde 3-phosphate are both produced. Well, there's an enzyme, triose phosphate isomerase, that takes dihydroxyacetone phosphate, converts it into the more usable for glycolysis, glyceraldehyde 3-phosphate. Arguably, an isomerase is kind of the easiest type of enzyme to understand, because all that you're doing is you're taking one molecule, one configuration of a series of atoms, and rearranging those atoms. As long as you know what an isomer is, well, an enzyme or an isomerase is a little bit easy to track what it's doing. You're rearranging the atoms to form a new molecule. The second to last class of enzymes are your ligases. Now, your ligases, what they are going to do is they're going to use the energy from the ATP hydrolysis to combine two molecules together. Now, an example of this is an enzyme known as glutamine synthetase. This is an enzyme that is involved in converting glutamate, an amino acid, a proteinogenic amino acid, into glutamine. Now, if you look at the functional group or the R group for glutamate, what you're going to notice is we have a carboxylic acid at the bottom of it. So that means we've got a carbon with a double bonded oxygen, a carbon with a single bonded oxygen to it as well. Now, our reactants are glutamate, ATP, and ammonium. So those three reactants are our inputs. Now, what glutamine synthetase is going to do is it's going to take that carboxylic acid and remove that single bonded oxygen and replace it with NH3+. Okay, so our ammonium is going to be added, oopsies. Our ammonium is going to be added to, well, that carbon at the end of our functional, at the end of our R group. Now, what are our other products? Well, our other products are ADP, adenosine diphosphate, and PI, inorganic phosphate. So what that's showing is our ATP, what happens was the gamma or the beta and the gamma phosphate, the phosphodiester linkage between the phosphoanhydride linkage between those two phosphate groups, that's broken. The energy released from that was enough for this ammonium to be added to my R group. So ADP and PI, well, those both came from ATP. The ammonium was kind of our target. The ammonium and the glutamate were intended to come together to make glutamine. So that's what a ligase is going to do. It's pretty common for you to see ATP as a reactant and then ADP and PI as your products. Now, our last group of enzymes, our last class of enzymes, are translocons. An example of a translocon is a sodium-potassium pump. Now, a sodium and potassium pump is a little bit complicated because of the fact that it's a multi-enzyme and a multi-subunit complex. But a little bit of a visual for what a translocon is going to do. Well, let's imagine that this is our lipid bilayer. Okay, so these two lines, these two horizontal lines, are indicative of the polar head groups, or what I'm trying to display there are the polar head groups for my lipid bilayer. In this region in here is where my aliphatic or my nonpolar chains are going to be found. Now, what a sodium-potassium pump is going to do is it's going to be moving sodium ions and potassium ions in opposite directions. Potassium ions are going to be moving into the cell, so interior, and outer. And sodium ions are going to be moving in the opposite directions as those potassium ions. So there's a lot involved in how a translocon, and specifically the sodium-potassium pump, functions. But at the end of the day, what's happening is two different ions in multiple, they're moving two potassium ions from the outside to the inside of the cell, and three sodium ions from the inside to the outside of the cell. So at the end of the day, what we're doing is we're moving these two ions in opposite directions as one another. We're going to try to increase the concentration of our sodium ions on the opposite side as our potassium ions. So translocon is a pretty big class because of the fact that it's multiple different enzymes and multiple substrates that are involved in that whole process. But that entire complex is referred to as a translocon and ends up as that seventh classification. Well, I hope this was helpful for breaking down all of our different classes of our enzymes and thinking about subclasses and subclasses, then ultimately our specific enzyme. Now, when it comes to knowing the class, subclass, subclass, and specific enzyme number, it's pretty uncommon that you would be expected to know all of them. Knowing what they are, they're kind of addresses. I always kind of think of them as addresses or similar to IP addresses, but for specific enzymes. All right, well, I hope you have a good one.